I can't go reward, only forward.
Well, yes, it says back. You can go with the red button here.
Oh, okay. Okay, thank you. Well, thank you. First, I want to thank Professor Magalon for this kind invitation. We worked together for years and years, and I am really happy to be there because I want to show for the first time to people generally involved in cells therapy and cells works, what is really the living matter. I am a surgeon, a plastic surgeon, a reconstructive surgeon. I was interested for many years in sliding system, and after many years of exploration of sliding systems, I will show you, I have been surprised by some cellular behavior. I was not really prepared to see and to observe and to think about this cellular behavior. But I did a lot of video sequence to try to understand. And so today I want to present not only the extracellular matrix, but also the relationships between extracellular matrix and cells in a living body, So, as a plastic surgeon, I did many liposuction, and of course, I know that I was destroying all these tissues under the skin, because you destroy the harmony of all these tissues. And of course, you destroy, you suck, you put it in a tube, and then you put all these cells in different chemical products, and then after you pigment, you enzymate, and at the end, are there really still cells? So this is a problem for me. But we have tried to make video concerning sliding system and cells everywhere in the body. Because I am a plastic surgeon, I had this opportunity to make all this exploration. And we did it with Professor Magalon one day. And we record all these images. We had a HD camera with an endoscope, with a cold light. It is an endoscope, a contact endoscope, with a zoom 25 times minimum. And of course, we are always, during some parts of the surgery, trying to make video with a focusing very difficult because the depth of field is very short, and you have very quickly many droplets of liquids, water, and you have to wash all the time to obtain a new world, a world of fibers, a world of colors, of sparkling mirrors, but in a complete disorder. This is the major observation. Very few Euclidean figures, but you have an irregular pattern. And I insist on this fact. We have an irregular pattern which is mobile, and fractalized. So the extracellular matrix is not at all what we have here. It's totally wrong. This way of thinking is absolutely simplistic. It's not the reality. So, the first, for a surgeon, when you incise, you are confronted to the basic physical factors. The basic physical factors are, first, some liquids. When your tourniquet is on, during, for example, forearm surgery, you observe liquids, lymph. these liquids, and if you put the tourniquet off, you have the blood, and then you can't make video because you have blood. And you are confronted to the systolic pressure, you can see there, and you can see here what is really systolic pressure. It's not nothing. When it's a big artery, it's a big problem. And when you make an incision, you can see the pressure pushing the fatty lobule upwards. And when you cut an aponeurosis, you can see that it's like a rubber band. And when you look at the ECG, you have electricity in your body. So you have a lot of forces. We are under forces. And when you observe that, you can see that a lot of fibers everywhere. There is a world of fibers. You call that extracellular matrix. But the extracellular matrix is a world. So have a look on the fatty tissue first, because we are interested by fatty tissue. When you put the endoscope inside of a living body, you see, of course, the fatty system, the fatty tissue. And when you make an incision, this is one of the first things you can see. This is the fatty lobules. And around, you have the extracellular matrix. But first, I want to say that there is a total continuity between the fatty system and the dermis. There is no free plane. This is an error. This is wrong. All is in continuity. All this plane, this free plane, don't exist. And you have a lot, if you use only 6x magnification, you can observe all these lobules. All of them are similar, but all of them are different. There is a total diversity in the shape, sometimes square, sometimes round, sometimes well vascularized, sometimes no. Look at the vascularization of a fatty lobule. And sometimes you have difference in colors. You can observe the traditional fatty lobule, yellow. But sometimes you can have fatty lobule, white. And you can see here. And the speaker just before me was talking about brown lobules. That is right. You can have white, you can have brown, you can have yellow. In the same place, in the same body, you can find brown, yellow, and white. And we don't know really what is the meaning of that. But it's a reality. And inside the fatty system, you have fibers. This is the main observation. Fibers everywhere between the fatty lobules. You have fibers at the level of the superficial fascia. Yes, of course. But you have fibers around all the fatty lobules. All of them are linked by fibers. The extracellular matrix is made of fibers. At all the levels you can find these fibers linking the fatty lobules. This is at the mesoscopic scale. And you can find all these fibers and with all these lobules in all the body, everywhere, forearm, abdomen, back, legs, everywhere. There is a sort of association at first glance between lobules, fatty lobules, and fibers. But if we go closer with the endoscope, we see that the fibers penetrate inside the fatty lobules. There is no separation between the connective tissue and the fatty lobule. Not at all. The fibers penetrate inside the fatty lobules. There is a total interpenetration. There is no connection. It's constitutive. So you can see these fibers, inside. And sometimes you don't have, like in this sequence, you don't have cells. Just beneath the fatty system, the hypodermis, you have this zone without any cells, almost any cell. Maybe you can find one. And this zone is the zone of gliding. When you have this, you are doing that, you are moving all this zone. And this zone is without cells. So the cells are not everywhere. So the cells are not the only responsible of the shape of a body. And this is important to know that. So what are the relationships between cells and fiber? Because we have fibers everywhere with cells. So have a look. First, we see that cells here are completely embedded by the fibrillar network. You don't have free cells. All the cells are completely nested, embedded in this kind of fibrillar network. You can see here cells, vessels, fibers, moving some. And if you go closer again with a magnification of 60 times, you can see that in some place you have no cells. Look here. And in other place you have cells. And the link between the fibers and the cells is total. Total. You can't separate a cell from the fibrillar network around. It's global. So, in the next step, next sequence, we are going to understand what is this fibrillar network. I have seen today and yesterday a speaker talking about the collagen fibers. Yes, but do you know that fibers are mobile inside the extracellular matrix? Let me show you. Look here. This is extracellular matrix. They move. All these fibers are moving. How are they moving? First, we have seen that it's an irregular network. But it's irregular, but there is no haphazard. There is a sense. The sense is suppleness. The sense is movement. The sense is force absorption. Look at the movement of a tendon. The tendon moves, but not the tissue around, because there is an absorbing system. It's the same here. This is a tendon, and this is the surrounding tissue able to absorb the force. So you have the tendon here, moving, efficient, and here, absorption of the force. How is it working to absorb the force? How is organized this fibrillar network, the extracellular world? It's organized thanks to intrinsic capacities of these fibers. I show you this sequence. Look, fibrils, look here. This fiber is going to move. The other one is going to divide. And during that time, the third one is going to lengthen. Again, sliding, dividing, and lengthening. In the same time, you have these three kinds of movement. And when you imagine this three kinds of movement on billions of fibers, you can explain the suppleness. And the suppleness you can have thanks to rings of elastin and collagen with retraction and lengthening, retraction and lengthening, but it's only one movement. The other one is a sliding of the fiber on other fiber. This is the second movement. And the third, this is an animation to explain. It's a sliding. You have here a sliding, look here, and sliding again. And you have also the division. Fibers can divide. And thanks to all this capacity, you have an explanation of suppleness and what we call elasticity. So in the 3 dimension of space, you have to add the fractalization. Look, 2, and suddenly 1, 2, 3, 4 fibers. So all these fibers are able to adapt to any constraint in the 3 dimension of space. This is how is working the fibrillar network, the extracellular matrix. Everything is under tension and everything is mobile, able to give a response to all the constraints in the 3 dimension of space. So we can say, what about the cells inside this network? The answer is almost simple. Here, you have the intercrossing of fibers in the 3 dimension of space. We know that it's an irregular network. But when you have fibers intercrossing in the 3 dimension of space, you have introducing the notion of volume. Volumes. And here, you can and we can explain little by little why we are volumes, how cells are arranged inside the extracellular matrix, because we have creation of volumes. And all the cells are nested in this kind of network. And this network is able to move. And the cells are using this network to be more adaptable, and we will see for more function. And the influence of the fibrillar network on cells is sometimes very brilliant. Look at the cells here. They are oblong, and suddenly they are becoming more circle. Here, cells are mobile. They move, they change of situation. So the mobility of cells is under the fibrillar network behavior. Now, what about the cells arrangement? Look here. The cells are along muscles, along tendon. Billions of cells without any order. There is certainly one, but impossible to understand. All these cells are covering arteries, veins, muscles, aponeurosis. Look here. It's like flowers in a field during springtime. You have cells everywhere. And in some part, no cells. And in some part, look here, you have a big group, and look on the side. Here, you have some cells completely a part of the group. What are they doing there? They are not in the group. Look here, another example. Are these cells migrating? Are these cells nomadic? This is the question. The same here. You have a group of cells here, along the vessel here, and when you go upwards, you have 2 cells more, 2 or 3 cells more. They are separate, they are not in a group. Here it's the same, a fiber and 2 cells. Another example, here, a fiber, again, and then suddenly 2 cells. Here, fibers, and a group of cells, all along the fibers. They are not in a big group, no. And here it's the same. Cells all along a fiber, and it is a fiber. Look, it's a fiber. So cells and fibers are very close, like here. So we decide to explore this zone between cells and fibers. What happened? Is it a complete link or not? And look, this is fiber, this is cells, and the fibers are disappearing inside the group of cells, inside the lobules. Totally. Look here, the junction, cells and fiber. So, and sometimes, I want to add, you have no cells along fibers. Why? I don't know. And so I was going on to explore cells, fiber. So it's a sort of pathway for cells. All these fibers are colonized by cells, like here. Cells, vessels, all along a fiber. It's not a fatty lobule, it's a fiber. Like here, you have a fiber with cells inside, with vessels inside, in a homogeneous fiber. So the links are total. You can dissociate cells, vessels, and fibers. One remark just before the conclusion. During our exploration, we have been surprised by this kind of cells. It's just before the aponeurosis, these sort of clusters of cells. They are not adipocytes. They are just beneath the hypodermis. Look here. This is a perfect example. It's a sort of cluster of cells, very well vascularized, and just before the pre-muscular or pre-aponeurotic area. Different kind of shapes. A small one here, completely nested in the fibrillar network. And these clusters are completely included in this fibrillar network. They are completely embedded inside this fibrillar network. Look here. And all these clusters are completely full of immature, of pluripotential cells. We have begun a study with our Society of Cellular Therapy in Bordeaux, and we have to go on. But here you have cells, but adipocyte, but you have a lot of immature cells, a lot of big nucleus, a lot of mitochondria, a lot of production of collagen. So all these clusters are really interesting to study. In conclusion, because I have not a lot of time, and that's okay. In conclusion, all this fibrillar network that I have seen during 40 years of surgery and 15 years of exploration of this extracellular matrix, this fibrillar network, which is found everywhere in the body, everywhere, in a muscle, at the level of the bone, at the level of the periosteum, between the fatty and inside the fatty lobules, at the level of the cells, with a role on the mechanical attitude of cells, at the level of the cells. And this fibrillar network that we can find not only in all the tissues, able to give the suppleness, the mobility, able to nest all the cells, found in all the anatomical zone, abdomen, extensor, scalp, thorax, back, everywhere. I found it everywhere. I explore it everywhere. This extracellular matrix, look here, between two muscles. This tissue, this network, network which has been neglected by all the anatomists and all the surgeons, and I am one of them. In fact, this extracellular matrix, this fibrillar network, I propose and I think that if we find it in the entire body, everywhere, at all the levels, could be our interior architecture, in our interior framework. Not the cells are our interior architecture. The extracellular network, the extracellular architecture is our architecture, interior architecture. And so you can define the skin, the bone, and the connective tissue is not only connective, it is constitutive. And it is a fundamental paradigm shift. You can't now think anatomy in the same way. We are in the globality. We are made of a global system. And we can define the skin, the fatty system, the sliding area, the tendon, the aponeurosis, the bone, even the bone, with the same architecture, the same framework. And we can have some design like that, with a total homogeneity, and plus able to resist to gravity, because we have to explain why we can be able to resist to gravity. So I invite you to a world of fibers with only one frame made of fibers and glycoaminoglycan for suppleness, elasticity, adaptability, mechanical adaptability, or the same framework with cells inside, with an operative and productive component, able to create glands, some secretory glands, and able to give also mobility. So this way of thinking anatomy and observing anatomy, I think, gives some solution for some pathologies and give also the opportunity to open new fields in many scientific fields, research, and could be a new way of thinking anatomy. Thank you very much.